Hi, my name is Brandy Leach. I'm a genetic counselor, and today I'll be talking through the genetic testing process and genetic testing interpretation with you. I am a full-time employee of Invitae, which is a genetic testing company. However, the principles I'll be discussing with you today are broadly applicable to any genetic testing process. In today's session, we're going to do an overview of cancer genetics. We'll discuss hereditary cancer contributions and principles, talk about next generation sequencing, outline the genetic testing process, and then talk through a sample test report. So I wanted to start with the central dogma of genetics. So genes are like a recipe or an instruction that tells your body how to grow and how to develop. In humans, we have 20 to 30,000 genes, and approximately 40,000 of these genes have been associated with human disease. So if you think about DNA like a cookbook, it tells us what can be made. And then through the process of transcription, the DNA is made into RNA, which tells us what will be made. And then through the process of translation, proteins, where the product itself are made. And then the collection of all of these proteins together are what make up our cells and tissues. When we think about cancer specifically, and we think about variants as they relate to cancer, all cancer is due to genetic variants or what we previously referred to as mutations. So a variant is simply a change in the DNA sequencing, a change in the DNA sequence from what we would typically expect to see. A somatic variant is a change that occurs in DNA after conception. So after the egg and sperm unite, and they had a typical sequence of the DNA. After that cell has then replicated, the DNA can acquire a variant or a genetic change. So this is a change that occurs within certain cells or tissue within an individual, and they cannot be passed down to that individual's offspring. So somatic variants that are acquired in key genes over time can lead to cells that become malignant. And when we think about cancer, there are two main types of genes that we think about. So one is tumor suppressor genes, where we would expect to see loss of function. So you can see in this representation of a cell, we have two copies of a gene, one from our mother, one from our father. And this individual starts out with two normal copies of that gene. Then over time, he or she acquires a mutation in one copy of the gene. And that mutation could occur due to, or that variant could occur due to any number of reasons, whether that's environmental exposures, lifestyle, or mistakes that occur as the DNA sequence is copied and replicated. And then over time, that second copy of the gene can acquire a second mutation or variant that then can lead to the potential for this cell to become malignant and eventually develop into a tumor. On the other hand, in proto-oncogenes, we can see that an individual can start with two normal copies of the gene, but when one variant or mutation is acquired in one copy or allele of the gene, that actually activates the gene. So it's like the gas pedal in a car, and then leads in a proto-oncogene to that cell potentially becoming malignant and developing into a tumor. When we switch then to think about genetic variants that are inherited or germline, what that means is that the DNA variant is present in either the egg or the sperm cell that created that individual. So that variant becomes present throughout that individual's body, including his or her egg or sperm cells. And these variants can be passed down to offspring. When there is a germline variant present, that means that fewer somatic variants are required for that individual to develop a cancer. So as you can see here, rather than in the last example with somatic variants, in the hereditary situation, this individual is starting already with one variant present in their two copies of the gene. So he or she only has to acquire one more variant in order for that cell to have malignant potential and become a tumor or cancer. So because of these principles that there's already one variant present, it's present throughout the body, we can see these individuals have an increased lifetime risk of developing multiple malignancies. We can see an earlier age of onset because, again, only one additional variant has to be acquired rather than in the somatic space, we need to see two variants acquired. And we can see risk to other individuals. So just to reiterate, in germline versus somatic variants, in the germline, these are mutations that are present in the very egg or sperm that made that individual, and they are present throughout that individual's body. They are heritable, so they can be passed down to the next generation. Overall, germline variants account for about 10% of common types of cancer. And it's important to test for these variants because it can have implication for future cancer risk for that individual. Versus somatic variants are variants that are isolated to the tumor or cancer itself. They are not present elsewhere in that individual's body, but variants are present in all types of cancers. And testing for these variants can have very important prognostic and therapeutic applications for that patient. There are many different reasons why it is important to test for germline genetic variants in an individual. So first, let's take a look at what it might mean for the patient. So this can be helpful for risk stratification for the patient to understand the risk of developing a future cancer and to use that information to guide how screening and surveillance should be used downstream for that patient. Also, for individuals who have a cancer diagnosis, germline testing in some situations can help determine if that patient is eligible for participation in a clinical trial or research studies, and can have important implications for decision-making for surgery, chemotherapy, targeted therapies such as PARP inhibitors or immune checkpoints inhibitors. So there's lots of benefits to the patient, but also for his or her family members, germline genetic testing can be important. Because if a variant is identified in a patient, we can then take that variant and test it in other family members to figure out who in the family also carries that variant and has an increased cancer risk and could benefit from increased screening and surveillance to prevent them from developing a cancer, versus relatives who might test negative for that variant. It would help us to understand that that individual then is likely at a general population risk of developing cancer and would benefit from doing general population-based cancer screening. In the United States, the decision about who should undergo germline genetic testing is largely driven by the National Comprehensive Cancer Network, or NCCN guidelines. So these guidelines outline a number of features that might be present in the patient's personal or family history to help us identify who should have testing. So for example, currently all individuals with ovarian and pancreatic cancer can benefit from germline genetic testing. Patients with early onset breast, endometrial, or colorectal cancer can benefit from testing, as well as individuals who might have striking personal or family histories of breast or ovarian cancer. So let's move on and talk about genetic testing itself and processes and important principles in genetic testing. And I want to take a moment just to differentiate genotyping and sequencing, especially if you're a provider who works with cancer patients and might be ordering genetic testing itself. So genotyping is when there is a genetic analysis completed that looks only at specific variants that are well known to be pathogenic and potentially associated with a disease or with therapeutic implications. So I tend to think of genotyping like looking at hotspot mutations in KRAS or BRAF that can have important therapeutic implications. So we're targeting where on the page in the recipe or where within the recipe we want to look for commonly known misspellings or variants. Versus sequencing itself is going to read through the entire page or the entire book to look for any misspellings or variants anywhere within the recipe. Traditionally, sequencing had been done with a technology called Sanger sequencing, but this has now commonly been replaced with next generation sequencing. In the germline cancer genetic testing space, what we are doing is looking for variants in cells without cancer. So this is different than somatic genetic testing, which is looking for variants in the cancer cells. We actually want to look at normal cells. And it's a process that's used to identify variants, either typically from a blood or a saliva sample. And the testing that can be performed is sequencing of one or more genes known to be associated with hereditary cancer syndromes. And in the setting of next generation sequencing, we tend to refer to these tests as panels. And there are many different ways that panels have been developed. So some of these might be gene focused, such as BRCA1 and BRCA2 alone. There are guidelines focused tests. So based off of things like NCCN guidelines that associate 8 genes with breast cancer, there may be a very targeted 8 gene panel that looks at breast cancer guideline genes. Or you can get more broad into tumor focused panels. So looking at any gene that has potentially been associated with breast cancer on a panel. And then there are much more broader pan-cancer gene panels where we are taking not only breast cancer genes, but also ovarian, endometrial, colon, pancreatic, prostate cancer genes, all aligned together on one test, looking for variation within any of those genes, even if it's not directly related to the cancer type that the patient has in his or her personal or family history. And what research has shown is that as your panel becomes more comprehensive, your positive rate of identifying variants increases. So when we look across the most common cancer types of breast, ovarian, colon, prostate, and pancreatic, and we do a minimal panel, so a smaller, for example, guidelines-based panel in the specific disease, our variant detection rate can range anywhere from 4 up to about 9%. When we expand to more intermediate sized panels represented by the orange bars, so this is looking at maybe now panels that have about 10 to 20 genes on them, the variant detection rate jumps up from about 9 to 15%. But then when we use comprehensive panels where we're looking at genes associated with multiple different cancer types on one platform, that variant detection rate goes up from about 14 to 21%. So the more genes you add onto the panel, the greater likelihood that you are going to find a pathogenic variant associated with a hereditary cancer syndrome. In terms of how the genetic testing process works, once a clinician decides that germline genetic testing is going to be ordered for a patient, selects which panel he or she is going to order and submits the sample to the laboratory. When that sample is received at the laboratory, the DNA is going to be extracted from the blood or saliva sample and amplified. It will then undergo a quality control check to ensure that there is enough quality DNA to run the assay. The DNA will then be targeted for the genes that were ordered by the clinician. This will undergo the next generation sequencing. And then the output from that sequencing will undergo a bioinformatics pipeline, which will help to identify if any variants were identified through the sequencing process, what those variants are. And then that information will undergo a variant classification process, which is a combination of technology as well as a team of scientists looking at those variants and the available evidence about those variants to make a determination about how that variant should be classified. Once that process is complete, a report is then written and resulted to the ordering clinician. On the back end, before a genetic test is even run, there are a lot of important technical considerations that a laboratory puts into place before designing an assay. So one is the laboratory is going to ensure that they are choosing a transcript of the gene that is associated with the disease in question. And in that transcript, they're going to want to ensure that they are looking at all exons, which are the coding regions of the gene represented by these blue boxes here, as well as minimally the immediate flanking intronic regions where there can sometimes be variants that affect splicing or how the introns get removed from the exons in the process of transcription. So there can be rare situations where sometimes variants associated with hereditary cancer syndromes can fall outside of exons or the immediate flanking intronic regions. So for example, sometimes there can be variants within promoters or deep within the intronic regions. So laboratories need to be aware if there are any variants associated with hereditary cancer syndromes that might fall in these regions outside of where typically we would have our primers, to ensure that an assay is developed to capture these rare variants in uncommon places. So some diseases or genes where we know that this is common. So in the APC gene, which is associated with familial adenomatous polyposis, we know that there can be some promoter variants that occur that can be associated with the disease in question. So for the APC gene, we know that analysis of the promoter needs to be included in the APC gene assay. And then for the NF1 gene associated with neurofibromatosis type 1, there are some well-described intronic or deep intronic variants in intron 1, 11, and 38 that have been associated with neurofibromatosis. So the assay for NF1 needs to ensure that they are looking at these deep intronic regions for these variants. When we think about the next generation sequencing technology itself, it performs very well with identifying single nucleotide variants. So these are variants that are missense, nonsense, or synonymous changes. So for example, changing out the letter A for the letter T in the genetic alphabet, or even areas of the gene where we can see small insertions or deletions less than 20 base pairs. Next generation sequencing does an excellent job of identifying any type of these types of genetic variants. There are some limitations, though, of next generation sequencing technology, and that is when we start to look at larger rearrangements. So large indels of 21 to less than 50 base pairs can sometimes challenge next generation sequencing technology. Complex rearrangements, so these are greater than 50 base pairs of insertions, deletions, duplications, or even inversions where the DNA sequence is actually flipped backwards, as well as copy number variants that might expand multiple exons, can be very challenging for next generation sequencing to pick up on its own. And that's where the bioinformatics pipeline that the sequence goes through after sequencing is complete is very important to help sort out these types of genetic alterations. In addition, other areas that can be challenged by next generation sequencing are areas that have repetitive DNA sequences, genes that might have pseudogenes. So there's perhaps a copy of a gene that is non-functional that carries a lot of similar nucleotide sequence to the functional copy of the gene, or variants that might be mosaic, so observed at a variant allele frequency lower than what we would expect. We can't always determine if those are mosaicism or clonal hematopoiesis. So this is where bioinformatics can, on top of the next generation sequencing technology, can help to sort out some of these challenges. And then a really important consideration is coverage. So this is important to think about both for germline as well as for somatic assays. And there are many different elements of coverage that we want to think about. So there can be breadth of coverage, so how much of your target is covered. For any given gene of interest, you want to see that greater than 99.9% of the exons are covered in the assay. You also want to know about depth of coverage, so how many reads align to each target. All target sequence should minimally have 50x coverage, but you really have a good quality read at an average of about 350x coverage. And then you want to know about the uniformity of coverage. So how uniform is that coverage between different types of samples or samples from different patients? So just to walk you through breadth versus depth of coverage, if you had 20x depth of coverage, you would expect that 95.9% of single nucleotide variants would be correctly identified and called by your next generation sequencing technology. But if you expand that depth of coverage to 50x, you would actually expect that 99.9% of single nucleotide variants would be correctly called. So if you have a higher depth of coverage, that reduces the gaps in sequencing due to difficult regions of the genome and increases your detection of copy number variants as well as mosaic variants. So just to walk you through here, this is your reference genome or gene sequence. These are fragments of DNA from that have been amplified from the DNA sequence from the patient. So at 1x coverage, you can see these fragments align, but there are gaps in where the fragments of DNA are covering the reference. But as you continue to add depth of coverage with additional fragments, you get overlap and close some of these gaps, so that you can see with greater depth of coverage, you have greater coverage of the reference genome or greater breadth of coverage. So a greater likelihood that you're going to find all of the variations present within the gene of question. So once the DNA has gone through this process of next generation sequencing and through the bioinformatics pipeline, one of the really critical elements is variant interpretation. So any two unrelated individuals have millions of DNA sequence changes that differ between them. And on average, 1 in 8 variants is observed one variant is observed every 8 bases throughout the exome. So a significant number of variants are present in every single one of us. Some of these variants change or the frequency differs by ethnic group. So what you want to ensure is, one, that the lab you're utilizing can identify the variants in the gene, but also that they can correctly classify if the variant that they've identified is actually associated with a hereditary cancer syndrome or a hereditary disease. Because as you can imagine, the vast majority of the variants that can be identified are actually benign and not associated with disease. So variant interpretation is the process by which we come to a variant classification. Variant classification is how we communicate connection to disease. So variants can be either pathogenic, associated with hereditary disease, or benign. But often, because of lack of research, the connection to a disease for any given variant can actually be uncertain. So the goal is that you want to accurately, efficiently, and transparently assess pathogenicity of a variant. And when there's insufficient information, you want to confidently conclude that the nature of the variant is currently uncertain. So we've looked to the guiding source on this, which is the American College of Medical Genetics and Association for Molecular Pathology, AMP guidelines, which have put together guidance on how variants should be interpreted based off of information from population databases, the nature or type of variant. So is it a nonsense or a missense? What sort of in vitro and in vivo data are needed? And what information can we glean from family studies to eventually interpret the pathogenicity or benign nature of a variant? So in the ACMG AMP guidelines, there is a 5-tier variant classification framework. So starting here on the left, if there is a variant with which an investigator or a scientist can look at the available data and determine with 90 to 95% certainty that that variant is unlikely to be associated with disease, it is given a likely benign or benign classification. So for most laboratories, if a variant is classified as benign or likely benign, these will not show up on a test report. These would usually be reported out as negative or normal results. On the other end of the spectrum, if there is a variant that there is available data that with 90 to 95, at least 90 to 95% certainty, we can say that it directly contributes to the hereditary disease, it is given a classification of pathogenic or likely pathogenic. So this would confirm a hereditary cancer diagnosis. And then as you can see, for the vast majority of variants, they probably fall within this uncertain category where there is insufficient evidence at this time to associate the variant with either being benign or with associating it with the disease in question. So when you get a result back, we really put them into these 3 buckets of positive, negative, or variant of uncertain significance. So a positive result means that a pathogenic or likely pathogenic variant has been identified. This can provide an explanation to the patient for why they developed their cancer. It can help to clarify if that patient might be at an increased risk of developing another cancer down the road. And it also can have implications for family members, as we discussed earlier. So if a pathogenic variant is identified in a patient, his or her family members might benefit from testing for that same variant to clarify their own cancer risk. It is important to note that a positive result does not mean that the patient will definitely develop a cancer. It gives us a range of risks for which that patient has of developing another cancer. This is also known as penetrance, so the likelihood of developing a cancer when any given variant is present. And there also can be variable expressivity, meaning we don't know how many features of the syndrome will occur in any one individual. So identification of the variant tells us the risk is there, but it is not definitive and absolute. A negative result means no pathogenic or likely pathogenic variant was identified. In this case, the medical management for the patient should be based off the patient's personal and family history. It does not mean that the patient has no risk of developing another cancer or a future cancer. And it's important to note that there are limitations to genetic testing technology. So not all assays can find all of the variants present within a gene. So for example, if years down the road, we find that variants within the promoter are actually associated with the hereditary cancer syndrome, but at the time we did the genetic testing, we weren't assessing the variant, we wouldn't have been able to identify that a promoter variant was present. So because of testing technology, because of things we are yet to learn through research are associated with hereditary cancer syndromes, We can never with 100% certainty say that a negative result rules out a hereditary cancer syndrome. And then with the variant of uncertain significance, or VUS result, we know that a variant is identified in the patient's DNA, but what we don't know at this time is if that variant actually is associated with the hereditary cancer syndrome, or if it is just a benign change. We often have to wait for more research to be done before we fully understand if there is any risk present to the patient. So for that individual who has a VUS, management should be based off of what is in his or her personal or family history and not based off of the genetic test result until that result can be reclassified as pathogenic or benign after additional research is done. So in our last few minutes together, I just wanted to walk you through a sample test report and things that you should be looking for when you get a test report back for your patient. So one, you want to ensure that the patient name and identifiers match up with the patient that you are working with. The test report should list the reason for testing and what test was performed. And then usually somewhere in the middle of the front page, you're going to see a call out of what was identified. So usually here you would see something like positive, negative, or variant of uncertain significance or uncertain. It should list out which gene the variant was identified in, if it's positive or uncertain. It should list what the variant is. Zygosity, was it present in one allele or in two alleles? And what classification was assigned to that variant. The report will also outline next steps. You can also see a clinical summary. So in this case, the patient has a likely pathogenic MSH2 variant. We know that pathogenic variants in this gene are associated with Lynch syndrome. So very high level summary of what Lynch syndrome is and the risks associated with it. You can also see variant details. So information that is known about the likely pathogenic variant that was identified in MSH2 in this patient. So what's in population databases, what functional studies have been performed, what we know about this type of variant, which in this case is a splice acceptor variant, and then how that information came together for the laboratory to make the interpretation that they did. The test report should ideally always list out the genes that were tested in the assay and provide some important information about the methods that the laboratory used for their sequencing and interpretation, as well as key limitations of the assay. And then a lot of laboratories now are providing additional guide reports. So in this case, this is an MSH2 guide, which talks about what disease MSH2 is associated with, which is Lynch syndrome, what the cancer risks are in association with Lynch syndrome, how Lynch syndrome is inherited, and then goes on the second page to outline what the current NCCN recommendations are for individuals who have Lynch syndrome, broken down by organ type, and what sort of screening or surgical or chemopreventive options are available to the patients. So in summary, at least 5 to 10% of cancers are due to hereditary cancer syndromes. There are another 10 to 30% who have familial clustering, so some combination of genetic factors as well as hereditary or genetic factors and lifestyle factors that come together to increase the cancer risk in the family. Germline genetic testing can have multiple benefits to the patient and his or her family members. The size of a gene panel is going to correlate with your positivity rate, and variants can be classified as benign, likely benign, uncertain, likely pathogenic, or pathogenic. And you need to be aware that for those uncertain findings, which occur relatively commonly, those really should not be used to guide the patient's care until further research can be done to clarify the significance of that variant in that patient. So I thank you so much for your time, and I look forward to our live Q&A session. Thank you.